Hello, good morning, all of you. So, we were discussing regarding the electron transport chain. The electron transport chain is a sequence of different carriers transporting or carrying electrons and protons, and by transportation of electrons, the energy in the form of ATP is generated. So, whenever there are dehydrogenations type of reaction, whatever protons or hydrogen are removed, they are transported along with electrons through the electron transport carriers, and at different steps energy in the form of ATP will be releases. So that transportation and organization of electron carriers in electron transport chain we are studying now. Here in this particular slide, it is shown that the electron transport chain, how the carriers are arranged. In previous lecture, we have already discussed regarding the electron transport carriers including NAD, FAD or FMN, then coenzyme Q or quinone, flavoproteins and chain of cytochromes. And lastly, the molecular oxygen in case of aerobic organism, which will be the final electron acceptor. In case of anaerobic organism, other than oxygen, the electron acceptors will be either nitrate, sulfate, etc. So this type of discussion we have already studied in last or previous lecture. Now we are studying here how these components are arranged in particular sequence and how there is a electron transportation. And lastly, then we will discuss regarding the ATP generation. So in this slide you will observe that there is a sequential arrangement of the electron carriers. First carrier will be NAD, that is nicotinamide adenine dinucleotide. Whenever it is accepting protons, it gets reduced to NADH. The NADH donates its proton and electrons to the next carrier in the electron transport chain, that will be either FAD or FMN. FMN or FAD accept protons and electrons from NADH and get reduced to FADH2. Later on, the flavoprotein transport its protons and electrons to the next electron carrier, that will be coenzyme Q or quinone. Quinone get reduced to quinol, and later on these electrons and protons will be transported to the chain of cytochromes. We know that the cytochromes are arranged in specific manner: cytochrome B, cytochrome C, cytochrome A, A3, like that. And the cytochrome sequence, it is totally depending upon the absorption spectra of the cytochromes. So later on, whatever protons and electrons are there which are from the coenzyme Q or reduced form of the quinone, they will be transported to the chain of cytochromes. And lastly, if the organism is aerobic, then final electron acceptor is molecular oxygen. So lastly, the protons and electrons will be accepted by 1/2 O2, that is oxygen, and then it is converted into water molecule. So here in this diagram you can also observe that at 3 different steps ATP will be generated. So the first will be transportation from NAD to FAD. Then we know that coenzyme Q to cytochrome, and lastly cytochrome to cytochrome oxidase, there is energy generation. So 3 steps there is a generation of energy whenever NAD will be the primary electron acceptor. So flow of electrons is in specific manner. The respiratory electron carriers, they are arranged in complexes according to their redox potential. So depending upon their oxidation reduction potential, the electron carriers are arranged in specific manner, in specific sequence. The redox potential measures the affinity of carriers for electron, means how much the affinity is there. So depending upon the affinity for electrons, the carriers are arranged in specific sequence, or you can say that these carriers, depending upon the absorption spectra, they are arranged in specific sequence. All carriers are arranged in specific sequence depending upon the redox potential. The complex one, or you can say the electron carrier, the first component will be NAD in electron transport chain. We have already discussed that. So NADH means reduced form of NAD. NADH quinone oxidoreductase or NADH dehydrogenase. Dehydrogenase is the enzyme catalyzing dehydrogenation reaction in which the hydrogen will be removed off and that will be accepted by NAD electron. So it consists of FMN flavoproteins and FES protein. FMN means flavine mononucleotide. FES means iron sulfur proteins. So this complex transfer hydride ion form NADH and proton from matrix to quinone. So NADH means reduced form. Firstly, NAD accept protons or electrons. So NAD get reduced to NADH. NADH transfer or donate its proton electron to FMN or FAD. Okay, and FMN or FAD get reduced to FMNH2 or FADH2. Later on, it is going to transfer electrons and protons to next carrier, it is the quinone. So quinone get reduced to quinol. So in this way, there is a transportation, means one carrier whenever donates the protons and electrons, it itself get oxidized, and the next carrier which is going to receive the or which is going to accept protons, it get reduced. Acceptance of protons, reduction. Donation of protons is oxidation. So there is a simultaneously or alternatively, you can say, oxidation and reduction. One component get oxidized, next component get reduced. The component who is going to donate electron and proton get oxidized. The next component which is going to accept protons and electrons get reduced. So there is oxidation, reduction alternatively. The electron transfer process is exergonic and releases energy. It is used to pump 4 protons from the matrix into periplasmic space. So there is a energy generation process. It is an exergonic type of reaction in which energy will be generated. So during the proton and electron transportation, at 3 steps energy will be generated here. The complex 2 in electron transport chain represent succinate dehydrogenase. It is the membrane bound enzyme from TCA cycle. We know TCA cycle very well. Succinate dehydrogenase enzyme catalyzing dehydrogenation reaction from succinic acid to fumaric acid. Succinic acid CH2COH, CH2COH. Succinic acid undergo dehydrogenation reaction whenever 2 hydrogens are removed. So there will be CH-COH double bond CH-COH, fumaric acid. So succinate to fumarate. This reaction will be catalyzed by enzyme succinate dehydrogenase. It is a membrane bound enzyme from TCA cycle. It extracts 2 electrons and 2 protons from succinic acid and forward to quinone through FAD. So first NAD, then FAD, then quinone. So there is a transportation of protons and electrons through FAD to next component or next carrier, that is the quinone. Quinone, it acts as a pool for electrons. The fully reduced quinine QH2 means reduced form of quinone, transfers 1 electron at a time to complex 3. Complex 3 means next electron carrier. So quinone, it is acting as a pool for the electron and whenever it is going to fully reduce, the quinol is formed and this quinol is going to donate or transfer electron to next electron carrier, that is the chain of cytochrome. That will be the complex 3. So the complex 3 represent cytochromes, cytochrome B, C1 complex. Here you can remember that cytochromes are divided or categorized depending upon their absorption spectra. Cytochromes present in prokaryotic cell are different than the cytochromes present in eukaryotic cell. Those cytochromes present in prokaryotic cell, they will be abbreviated as cytochrome B, C, A, A3 like that. Sometime in case of eukaryotic BK, BT like that. So you should think that in case of eukaryotic cell, that is electron transport chain is located in mitochondria, because mitochondria is there in eukaryotic cell, but it is absent in prokaryotic cell. So you can remember that in case of bacterial cell or in case of prokaryotic cell, electron transport chain is located in the cytoplasmic membrane. So cytoplasmic membrane is the major site for ATP generation in case of prokaryotic cell. So here the cytochrome B, C, this constitute complex 3 in electron transport chain. So this consists of cytochrome B as well as iron sulfur proteins and cytochrome C1. So cytochrome B, cytochrome C, cytochrome A, A3, these are the different types of cytochromes, and in case of prokaryotic cell especially. So its main function is to receive one electron at a time from quinone. Previous electron carrier will be quinone. Quinone, when accept proton, it get converted into quinol. Quinol, quinol. Quinone to quinol. Quinol is reduced form. QH2 means it is the quinol or reduced form of quinone. So QH2 get oxidized. When it will get oxidized? When it is donating proton to cytochrome. QH2 do not donate proton and electron to cytochrome. So quinol get again converted into quinone. So QH2 get oxidized to quinone only. Quinol to again quinone. Quinol will be reduced. Quinone will be oxidized. And 2 molecules of cytochrome get reduced. So here we should remember that in cytochrome, iron is there. Iron, or you can say here it is the prosthetic group, iron or heme. It is tightly bound to the moiety or cytochrome structure, and iron, it may be present in ferrous or ferric form, Fe plus 2 and Fe plus 3. Fe plus 3, that is the ferric form. Fe plus 2, that is the ferrous. Ferrous means reduced, ferric means highly oxidized. So cytochrome C is soluble protein of periplasmic space and transfer electrons to complex 4. Next complex. The complex 3 म्हणजे सायटोक्रोम बी, आयर्न सल्फर सेंटर, वेगवेगळे कॉम्प्लेक्स आपण बघितलेले आहेत. NAD, FAD आणि आता सायटोक्रोम आणि आयर्न सल्फर प्रोटीन, हे वेगवेगळे कॉम्प्लेक्स आहेत. या कॉम्प्लेक्समधनं इलेक्ट्रॉन ट्रान्सफर होत असतात आणि इलेक्ट्रॉन आणि प्रोटॉन ट्रान्सफर होत असताना 3 स्टेपमध्ये एनर्जी जनरेशन होत असतं. तर पहिला जो कॉम्प्लेक्स आहे तो आपण NAD पाहिला, नंतर FAD आहे आणि मग क्विनोल आहे. तर क्विनोन जेव्हा इलेक्ट्रॉन किंवा प्रोटॉन एक्सेप्ट करतो तेव्हा क्विनोनचं कन्वर्जन क्विनॉलमध्ये होतं, QUINOL. तर क्विनॉल किंवा QH2 असं इथं दाखवलेलं आहे. तर QH2 म्हणजे रिड्यूस फॉर्म ऑफ क्विनोन. तर क्विनोन गेट कन्वर्टेड इंटू क्विनॉल, सो दॅट इज द रिड्यूस फॉर्म, अँड आफ्टर देन the electrons are transported to the next complex, that is the complex 4. The complex 3 works as a proton pump and pumps 4 protons for every pair of electron transfer. म्हणजे इलेक्ट्रॉन ट्रान्सफर होत असताना सायमल्टेनियसली प्रोटॉन पण ट्रान्सफर होत असतात. तर 4 प्रोटॉन प्रत्येक इलेक्ट्रॉन पेअरबरोबर ट्रान्सफर होत असतात. सो द कॉम्प्लेक्स 3 वर्क ॲज अ proton pump and it pumps 4 protons and for every pair of electrons which are transferred. Then lastly the complex 4, it is representing cytochrome oxidase, cytochrome C oxidase. तर सायटोक्रोम C आहे, परत सायटोक्रोम A आहे, सायटोक्रोम A3 आहे आणि सायटोक्रोम A3 together it is called as cytochrome oxidase. So it consists of cytochrome A and A3. A आणि A3 मिळून त्याला काय म्हणायचं, सायटोक्रोम ऑक्सिडेज. It receives electrons from cytochrome C and transfer it to cytochrome A3. सायटोक्रोम C कडून इलेक्ट्रॉन ट्रान्सफर होत असतात सायटोक्रोम A आणि A3कडे. अँड फायनली सायटोक्रोम्स विल बी ट्रान्सफरिंग इलेक्ट्रॉन्स टू टर्मिनल इलेक्ट्रॉन ॲक्सेप्टर विच इज मॉलेक्युलर ऑक्सिजन. जर ऑर्गॅनिझम एरोबिक असेल किंवा एरोबिक रेस्पिरेशन जर असेल तर फायनल इलेक्ट्रॉन ॲक्सेप्टर हा ऑक्सिजन असतो. आणि जर ऑर्गॅनिझम अनएरोबिक असेल तर फायनल इलेक्ट्रॉन ॲक्सेप्टर हा अदर दॅन ऑक्सिजन म्हणजे सल्फेट किंवा नायट्रेट यापैकी असू शकतो. सो देर विल बी ट्रान्सफर ऑफ इलेक्ट्रॉन्स फ्रॉम सायटोक्रोम A3 टू टर्मिनल और फायनल इलेक्ट्रॉन एक्सेप्टर. ॲज रिझल्ट द मॉलिक्युलर ऑक्सिजन गेट रिड्यूस टू वॉटर मॉलिक्यूल. आता या ठिकाणी काय होणार, नुसते इलेक्ट्रॉन ट्रान्सफर होत नाहीत तर प्रोटॉन पण ट्रान्सफर होत असतात म्हणजे H+ म्हणजे प्रोटॉन, E म्हणजे इलेक्ट्रॉन. तर प्रोटॉन म्हणजे H+ हे जेव्हा इलेक्ट्रॉनबरोबर ट्रान्सफर होत असतात तेव्हा त्याची रिॲक्शन 1/2 O2 म्हणजे मॉलिक्युलर ऑक्सिजनबरोबर होत असते. 1/2 O2 + H म्हणजे काय होणार, H2O म्हणजे वॉटर मॉलिक्यूल त्या ठिकाणी तयार होतो. जर मॉलिक्युलर ऑक्सिजन हा फायनल इलेक्ट्रॉन ॲक्सेप्टर असेल तर इफ मॉलिक्युलर ऑक्सिजन विल बी द final or terminal electron acceptor, then proton will be going to react with 1/2 O2 and then there is a formation of one water molecule. So 4 electrons and 4 protons are needed to reduce molecular oxygen to water molecule. The electron transfer take place at a time without leaving incompletely reduced intermediates such as hydrogen peroxide or hydroxyl free radicals. आता काही वेळेला कसं होतं की H2Oच्या ऐवजी काही वेळेला हायड्रोजन पेरॉक्साईड पण तयार होऊ शकतो, आयदर H2O2 वगैरे. पण H2O2 हा जनरली कसा आहे, डेट्रिमेंटल आहे, लिदल आहे ऑर्गॅनिझमना. H2Oच्या ऐवजी काही वेळेला H2O2 पण तयार होऊ शकतो. तो H2O2 जर तयार झाला म्हणजे हायड्रोजन पेरॉक्साईड तर ते सेलला लिदल आहे, डेंजरस आहे किंवा हार्मफुल आहे. तर ज्या बॅक्टेरियल सेलमध्ये इन्स्टेड ऑफ H2O2 जर तयार होत असेल तर सेल काय करते, सेल इज प्रोड्युसिंग एन्झाईम कॅटालेज अँड कॅटालेज विल ॲक्ट ऑन H2O2 और हायड्रोजन पेरॉक्साईड अँड स्प्लिट H2O2 into H2O and O2. In this way the lethal effect can be avoided which is due to H2O2. पाण्यामुळे होणार नाही पण H2O2मुळं लिथल इफेक्ट सेलवरती होऊ शकतो. तो इफेक्ट टाळण्यासाठी सेल काय करते, एन्झाईम तयार करते कॅटालेज. आणि ते कॅटालेज एन्झाईम H2O2वरती ॲक्ट झाल्यानंतर H2O2चं स्प्लिटिंग होतं आणि H2O आणि O2 तयार होतो. त्यामुळं सेलचं प्रोटेक्शन त्या H2O2पासून होऊ शकतं. दिस इज वन मेकॅनिझम हिअर. बट मोस्ट ऑफ द टाईम वॉटर इज प्रोड्यूस. बऱ्याच वेळेला फक्त वॉटरच तयार होतं, काही वेळेलाच H2O2 तयार होतो. सो हिअर दिस इज रिगार्डिंग द sequence of electrons or flow of electrons through different complexes and how the protons will be transferred from one component to another component and how there is a electron and proton transportation. So this is regarding the metabolism or this is regarding the electron transport carrier. Thank you.